Good day, this is Professor Will, CBMD PhD, and today's Thursday, December 16th, 2021. It is 7:03 and 17 seconds, p.m. Eastern Standard Time. Today's research, CRISPR-Cas9. So let's hop to it here. Breakthrough in using CRISPR-Cas9 to target fat cells. The method can help speed genetic discovery in the study of obesity. Date December 15th. 2021. Source: Michigan Medicine, University of Michigan. Researchers describe a breakthrough using CRISPR-Cas9, a tool that has transformed molecular biological research, but whose use in the study of adipose tissue had been elusive. Fat cells are different from other cells in that they lack unique cell surface receptors and only account for a minority of the cells within fat tissue. The biggest challenge in terms of adipose research to date has been that if you want to study a gene function, you have to commit a considerable amount of time, resources, and money into developing a transgenic mouse. The traditional way of developing mouse models involves breeding mice with a desired mutation to delete or introduce certain genes of interest, which can take more than a year and tens of thousands of dollars. CRISPR-Cas9 has revolutionized this process. It is a gene editing technique comprised of an enzyme called Cas9, which can break strands of DNA and a piece of RNA that guides the Cas9 enzyme to a specific site in the genome for editing. And this tool is packaged into a non-harmful virus for delivery to the cells being studied. The tool has been successfully used to study heart, liver neurons, and skin cells, to name a few, but never a certain type of adipose cells, known as brown fat. And using the technique, the team was able to successfully target brown fat, a specialized adipose tissue, used to generate heat and protect core body temperature. What research has been able to do is to take the whole process and distill it into anywhere from 2 weeks to a month to generate a transgenic mouse, reducing the cost to less than $2,000. Not only does it reduce the time and cost, it democratizes the research so that any lab that is familiar with molecular biology techniques can adopt this method and do it themselves. They were also able to use this method to delete multiple genes simultaneously, a fact that could help researchers better understand important molecular pathways. Using their adeno-associated virus CRISPR-Cas9 components, they were able to knock out the UCP1 gene that defines brown adipose and enables it to generate heat in adult mice. They observed that the knockout mice were able to adapt to the loss of the gene and maintain their body temperature in cold conditions, hinting at other pathways involved in the temperature homeostasis. And these early results are exploratory, but the technique represents an important step forward in studying fat. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.